Thanks for a reminder, by the way. So, I told AP Biology the other day, probably the most amazing thing about biology when you break it down is the fact that everything, life is a code. Everything is made up of a code. So your DNA, it's a code. Every protein in our body is made using a code. And to me, that blows my mind. Probably a very close second is the cell membrane. So whenever you hear cell membrane, you're probably taking notes, or they're talking about something called the plasma membrane. We do this from your sophomore year, it becomes a cell membrane. Now it's going to be a plasma, same thing. Same exact thing. Okay? This thing is absolutely amazing. Okay, so if you remember, this is what we should be covering today. Cells have membranes, allow them to establish and maintain internal environments that are different from their external. Remember, whatever's going on in that cell, you may not want it mixing with what's going on outside. And this really flimsy bubble is what keeps that from happening. And I don't know if I told you guys, but bubbles that you make with soap are very similar. Okay, just it's the way that the molecules are arranged in a soap bubble versus a cell. Now, there's this idea that it separates, right? One cell from another. Maybe what's happening outside, you might say this might be the outside, this might be the inside. It's going to separate what's happening here from here. Now, there are going to be, always going to be aqueous environment. Aqueous just means H2O, water. All of our cells are kind of bathed in this watery fluid. And so you have one layer, and this big, thick inside layer, and then the third layer. And always kind of keep in mind that a cell membrane is actually, it's made up of, they call it two layers, and you can see here's one, here's another, but there's three distinct behavioral layers. Okay, so this part here loves water, so it'll turn toward water. Here's another one that loves water, so it'll turn toward water. But these things that look like legs or tails, they don't like it, so they will turn away. And like I always tell AP kids, if you would take a handful of these things that are called phospholipids and you throw them in water, they will always arrange themselves in this way. Always. They love water, the heads will turn in, tails will turn away. Okay. So keep in mind, water will not be here. Polar, non-polar. These tails are fat, so water can actually seep through the membrane, but it won't stay there because water is not welcome there. Now I want you to draw this little double-tailed egg. This is called a phospholipid. This is called a phospholipid. And probably for you guys, the most important thing to know is that the head of it is polar. So probably most of the time it's got a slightly positive charge. It's polar. So if there's a negative charge anywhere, it's going to draw it in. Well, water sometimes has water molecules all the time have a negative end, so they will attract the water. But the tail is is that lipid part. This would be the phospho part. This is the lipid part. Remember, lipids are fats, waxes, and oil, and they hate water. And so, non-polar, since water's polar, it won't be attracted here. Okay, so their form fits the function. Their form fits their function. Okay? And you can see in this picture that look like they're yellow to orange head with tails. And you can see there's lots and lots and lots of them. But they're not the only thing in that membrane. There are other things. There are proteins that are studded inside that. Some are built in, some float on the top, some float what you might consider on the inside or some on the outside, at the top, the bottom, however you want to look at it. The ones that are peripheral are the ones that cling I don't know. They don't penetrate the membrane. So right here is a peripheral. Periphery means it's on the outside. That's one. You see that it doesn't go all the way through. Chances are this one here is probably one, as far as I can tell. So that's peripheral proteins. And they have a lot to do with cell communication. So like if you need a signal sent through the cell, these are the guys that you go to do it. Okay? Peripheral. So think about on the periphery, on the edge. That would be, I guess, on the edge. The integral. I always think of it like this. They're integrated. They're part of the membrane. This could be one. Here's another one right there. You can see that they span the membrane. They go all the way through it. Okay? So when you hear that, just know it's integrated and it spans. Okay, the membrane. Peripheral on the edge, integral, they're integrated. So they will actually have a section like this. This would be a non-polar side. This would be a polar, polar. And since they're arranged that way, look how they attach. That's pretty cool. But this is not what blows my mind about this. When you think of soap bubbles, do you think strong? No, they're not. And so they're not strong at all. In AP, we do what's called a bubble lab. And I add some corn syrup that makes the bubbles last longer, makes them tougher. It's really cool. All right. Look at some of these functions of the proteins. So this could be your peripheral, remember, and this could be some of your integral. This is their basic functions. We don't go into these too awfully heavy. When we get into cell communications, we'll talk a little bit about it. Okay. Transport. So they're going to allow some things into the cell. Remember I told you water has difficulty going through. All right, the inside here, this second layer here, doesn't like water. So when water does trickle through, it gets out. You see this protein right here? There is a protein made just for water. Nothing else goes through it. It's just water. It's called aquaporin. And it's a protein that spans the whole membrane. And so when you get thirsty and you take a drink of water, the water is bathing the outside of the cell and it needs to get through. This is the channel which it will start leaking through. Okay. And it's just not it's not pumping it. It's just behaving as water does. Water moves through on its own, requires no energy. And so it will leak just through that. Cell recognition. See this one right here that looks like it's got an antenna? That's called a glycoprotein, because it's a protein that we know it's embedded in, but you can see there's a sugar, glycogen, attached to it. That's cell recognition. So if we have white blood cells that are constantly going around feeling every cell in our body to make sure it should be there. If it's not there, then it turns into an attack dog and takes it out. So what it's feeling for is this. It might feel this and say, oh, okay, you're supposed to be here. But even if this is even one off, let's say that one's gone, it'll recognize it and say, oh, wow, okay, you don't belong here. And it'll start tearing it up. It'll kill that cell, whatever it is. Okay, signal transduction, probably this one joins one cell to another. Probably that one right there. And so these cells will get locked in, right? And so it's like, you remember that, what's that when you were a little kid, you all joined up, I did it too. We joined elbows and, was it Red Rover? Send somebody over, you ever play that? Yeah, and so these are interlocking, right, for strength. All right. Any questions so far? Remember, kind of know them by sight. Peripheral, integral. Okay, now, last one. If I say cholesterol, bad or good, what do you think? What's your first reaction? not very good. If you get too much of this, it could be bad, right? So if you look at it here, see that green structure right there? This would be a cholesterol molecule. It's not really how it works or looks, but they put it in there so you can see. The whole idea between cholesterol is it stiffens the cell membrane. Now there's a little more to it, but you don't need to know it. That's for AP. But just know if you get too much of it, the cell membrane gets too stiff. And then people that strain, you know, when your blood pressure, you're lifting stuff, sometimes you'll have a brain bleed. Your blood vessels can't contract and expand properly, and so you might get a brain bleed. So this is why they don't want you eating too much cholesterol, but I don't know. So it stiffens the cell membrane. So it's very, very, very important. Okay. Now, here's the cool thing. This is what blows my mind. Remember, each one of these have an slightly electrical attractions. Most of the time they're positive, but sometimes they can be negative. They are constantly shuffling places with one another. So when you blow a bubble, you ever notice the colors on it? Do they just stand still? They don't, do they? If you like blow a little wind on them, it will move. And so you can actually see their surface moving. And so that's what makes bubbles a lot like cell membranes, because these are constantly switching places with each other. It's always in motion. The picture doesn't do it justice. If I could find one, you would see these things switching places. They would be constantly in motion. Every once in a while, one will flip and trade, but that's not very often does that happen. Now, that blows my mind, because that means that every cell membrane layer is just moving. Can't feel it. It's kind of disconcerting. And it's very fragile, like a bubble. But anyway, they call that the fluid mosaic model. Okay, that's the latest trend. Matter of fact, that hasn't changed in about 15 years. We've been teaching about the fluid mosaic model, maybe. Okay. It's fluid because it's constantly moving. It says this is a bilayer, phospholipid bilayer, and it says it's viscous. You know what the word viscous means? It's thick in consistency. It's not like a liquid. It is a liquid, but it's thick. And so it can move all around. So each one of these are trading places all the time. And so that's why they say it's fluid. Now, mosaic, if you know anything about art, right? If you make a tile mosaic, are you going to use the same color? Probably not. You're going to use the same tile shape? Yeah, maybe not. You're just going to be making it of a bunch of different stuff. And so it's a fluid mosaic because it moves, and it's made of lots of different parts. So that's what they call that. So whenever you hear fluid mosaic model, remember, this is a current theory, but it's a pretty good one, because we can see it on bubbles happening. Right? And then how everything moves and everything is in it's different. And I think that's it. All right.